Good morning and welcome back to Open Med School. Today we'll be discussing about action potential. So, what is an action potential? Action potentials are brief, rapid, large propagatory changes in the membrane potential when adequate stimulus is applied to an excitable tissue. So, action potential travels as an impulse. This will be discussed in this session. So, what are the changes occurring in an action potential? There is depolarization and repolarization of the membrane. But this is a rapid, brief, large propagatory change. That is the action potential. So, to learn this, we should recap what is membrane potential, because action potential is a large transitory change in the membrane potential. So, as we know, the membrane is a bilayered lipid one with proteins inside that are acting as a channels, transmembrane proteins. So, there is a transmembrane, these are some of the channels there. It contains glycolipid, that is lipid with carbohydrate, and glycoprotein, that is protein attached with the carbohydrate and cholesterol. So, this is the basic structure of the membrane. And the membrane has got potential, and the outside of the membrane is positive and inside is negative, and that is what is called the resting membrane potential. The resting membrane potential for a nervous tissue is approximately 70 milliequivalents, that is minus 70 milliequivalents. That is the for nervous tissue. Other tissues it differs. And again, you should remember that the outside contains more of sodium and inside contains more of potassium. So, potassium is the intracellular cation, sodium is the major extracellular cation. And this is maintained mainly by the sodium potassium channel. And now we look into what is depolarization, what is repolarization, what is hyperpolarization. So, we know the membrane has got inner aspect is, inner side is negative and outer side is positive. This is what is called the polarized state. When this gets reversed, it is called depolarization, and then when it comes back to the same stage, it is called repolarization. So, a membrane is polarized. Polarized means outside is positive and inside is negative, and these changes are mainly occurring due to the sodium channels which becomes activated and deactivated. So, you can see a gate here, and some of the channels are activated by voltage, and such channels are called voltage gated channels. Voltage gated channels are there for sodium, potassium, calcium, and various other ions also. So, let us see where action potential occurs. Action potential doesn't occur in all the tissues. It occurs, it is one of the peculiarity of the excitable tissues. Which are the excitable tissues? The nervous tissue and the muscles are the excitable tissues. So, now we look into the action potential in the neuron. Same type of action potential can occur in cardiac muscle, the smooth muscles, and some of the glands also. So, we are mainly concentrating on the generation of the action potential in the nervous tissue. So, this is the typical neuron which has got an axon, the long part, this is the axon, and small, this is the dendrites, there is a cell body, there is nucleus, etc. And this terminal part of the axon is called axon terminal or axon button. And this portion of the neuron where the axon is connected with the cell body or soma is called the axon hillock. Axon hillock is very important because many times the action potentials arise in the axon hillock. And some of the neurons are having a covering, that covering is called myelin. When the nerve is having a myelin, it is called myelinated, and where there is not much myelin, it is called no myelin, it is unmyelinated neurons. So this is how we have said that the action potential travels as a wave and forms or it acts as a impulse. So the action potential travels only in one direction depending upon the neurons. So in the axon, it is here, you can see that is going in this direction, and in the dendrites going in this direction. So this is not a byway, it is only in one direction it travels, and direction depends upon the type of neuron, whether it is a motor neuron or a sensory neuron. It varies. And now look into what is the role of action potentials in neurons. So why should there be action potential? We have said that the action potential travels, and the traveling is like a wave. So from the cell body via the axon, the impulse is traveling down. Once the action potential reaches the terminal button or the axon terminal, there are vesicles containing chemicals there. These chemicals will be released into the synaptic cleft. So actually the action potential is used to communicate with the end organs or target organs. So here you can see it goes to the synapse. In the synapse there is a chemical transfer of information to the next tissue. So this is the function of the action potential. Now we see how the action potential is generated. Now let us see how an action potential is generated in the neuron. Here you can see that the neurons are receiving impulses from different areas or from different neurons. So here is one neuron, there is another neuron, this another neuron, all are communicating with this neuron. So when an impulse comes, some of the impulses are stimulatory or excitatory, and that produces a change in the membrane potential. So here you can see there is a change in the membrane potential because of this stimulus sensitive sodium channel, sodium is entering. But this is insufficient to produce or generate an action potential. But suppose you see that all these occurring together, or it is summated, that is called a spatial summation. At the same time, all the neurons are giving the stimulus, excitatory stimulus. What will happen? This will reach a particular membrane potential, that it is minus 55. This minus 55 is a threshold potential at which an action potential can start. So this is what we have mentioned earlier. Action potential is a transitory change in the membrane potential which is rapid and sudden, and it is propagatory. These impulses originating together is called spatial summation, or the impulse can occur repeatedly together so that this may reach the target threshold potential, that is called the temporal summation. So whatever way the neurons reach, the membrane potential reach minus 55, then there is a sudden opening of the sodium channels which are voltage sensitive. So what will happen? This will overshoot and it may reach up to minus 61, which is the Nernst potential for sodium. And once the potential reaches plus 30, what happens? The sodium channel gets deactivated or it gets inactivated, and the potassium channel opens. So what will happen? The potassium channel, the potassium will go out of the cell. So the positive charged potassium, because the inside of the cell contains more potassium, so the potassium channel opens and potassium goes out of the cell. This will reduce the positivity inside the cell becomes negative. So that is the beginning of the repolarization. So what happens? The potassium channels remain open for some more time. So even when the membrane potential is reached, it will go down. And that is called the hyperpolarization. Hyperpolarization means the membrane is more negative. So more stimulus is required to get it stimulated. That is what is hyperpolarized. Once it is hyperpolarized, it again go back to the normal resting membrane potential by the actions of the sodium potassium pump and other leaky channels. So this is in short the mechanism of production of action potential in a neuron. As I already said, there are inhibitory neurons also. The inhibitory neuron, what happens? They make the membrane hyperpolarized. That is, it becomes more negative. Hope the mechanism of action potential generation is clear and is usually generated from the axon hillock. So this is a region where the impulse starts in the action potential wave starts is from the axon hillock. Now we look a little more into each part of the action potential. How does it look like? If you record with from a nerve fiber using microelectrodes, there is one electrode is inside the cell and the other electrode, so one is inside the cell, inside is negative and outside is positive. So you are giving a stimulus, so this is the stimulator and you are going to record it. So you can see this is the like the all of you take a bit of paper and draw it and learn, otherwise you will easily forget this. So this will be the membrane potential is minus 70, threshold potential is minus 55. It overshoots depolarization, may go up to the next potential for sodium, and the potassium channel opens and the repolarization wave occurs. The potassium channels remain open for some more time, so it becomes hyperpolarized and later it goes back to the normal resting membrane potential. Now look into what are the events or phases of action potential. So the x-axis is time in milliseconds and this is the showing the membrane potential. Minus 70, it is the resting membrane potential. 0 is so here you can see stimulus is applied. So there is this part gets a stimulus artifact. Then comes a latent period. So what is my latent period? The stimulus applied and the beginning of the change in the potential is the latent period. Then you can get a transitory change in the membrane potential because this is a graded potential occur or a local potential. It has not reached the firing level. Then once it reaches the firing level, that is the threshold potential, action potential is generated. So that is a action potential is the wave is due to the rapid depolarization. Then it reaches, it overshoots, it comes down. A rapid repolarization wave is due to the opening of the potassium channels. Then there is a after depolarization and the membrane gets hyperpolarized because of the opening of the potassium channels remaining for some more time, so it becomes more negative predominantly. Let us look into any difference of the action potentials in different tissues. The shape is different. In nerves and muscles, it produces a spike like what we have drawn. In cardiac muscle, it is a plateau. You can see the cardiac muscle, the action potential has got a plateau like this. And in the smooth muscle, it is a little more smooth. So this is the slow potential that is occurring in the smooth muscles. So in this session, we have discussed about the resting membrane potential, how the action potential look like, how it is generated, and what is the ionic basis, what is the purpose of the action potential, and how the shape of the action potential differs in different tissues. Hope it was useful to you. We'll come with the synaptic transmission in next session. Thank you.